Welcome everyone. I'm excited to kick off our presentation today on glycogen metabolism and gluconeogenesis. This is the first slide, and it sets the stage for what's to come. We'll be exploring how our bodies store and generate glucose, which is crucial for energy. Over the next few slides, we'll dive into the processes of glycogen synthesis and breakdown, as well as how new glucose is made when needed. I'm glad you're here, and I look forward to sharing this fascinating topic with you. Let's take a closer look at the structure of glycogen. Glycogen is a homopolysaccharide, meaning it's made up of only one type of sugar, which is glucose. Specifically, it's composed of branched chains of alpha-D-glucose units. These chains are connected primarily by alpha-1-4 glycosidic bonds, which form the main backbone of the molecule. However, at certain points, the chain branches off, and this is where alpha-1-6 linkages come into play. These branch points are critical because they create a highly branched, tree-like structure. Each branch typically contains about 12 to 14 glucose units, which allows for rapid mobilization of glucose when needed. Most of the glycogen in our body is stored in the cytosol of liver and muscle cells. This strategic location enables quick access to energy, whether it's for maintaining blood glucose levels or fueling muscle activity during exercise. Understanding this structure is fundamental because it underpins how glycogen is synthesized and broken down, ensuring our bodies have a reliable energy reserve. Let's take a closer look at the glycogen stored in the liver and muscles. Starting with the liver glycogen, it primarily comes from blood glucose, but it can also be derived from other hexoses like fructose, as well as non-carbohydrate sources. The maximum amount stored in the liver is about 120 grams, though this can vary depending on the liver's size. The concentration is roughly 6%. The main role of liver glycogen is to serve as a backup source of blood glucose, especially vital for the brain during times of need. After fasting for about 12 to 18 hours, liver glycogen stores are usually depleted. Now, moving to muscle glycogen, which is mainly derived from blood glucose, and similar to liver glycogen, it can also come from fructose. The maximum stored amount in muscles can reach around 400 grams, depending on muscle mass, with a concentration of about 1%. Unlike liver glycogen, muscle glycogen isn't used to maintain blood glucose levels. Instead, it's a direct energy source for contracting muscles and helps produce lactate during intense activity. When glycogen is broken down through glycogenolysis, the end product in the liver is glucose, which enters the bloodstream to supply energy throughout the body. In muscles, the end product is lactate, which can be used elsewhere for energy or converted back to glucose in the liver. Regarding hormonal regulation, adrenaline, noradrenaline, and glucagon all stimulate glycogen breakdown, glycogenolysis, and inhibit glycogen synthesis, or glycogenesis. Insulin, on the other hand, has the opposite effect, promoting glycogen formation. Interestingly, glucagon does not have a significant effect on muscle glycogen metabolism, only affecting the liver. So, overall, glycogen metabolism is tightly regulated by these hormones to ensure energy availability matches the body's needs, whether for maintaining blood glucose or powering muscle activity. Let's talk about glycogen metabolism, specifically the synthesis of glycogen, or glycogenesis. This process involves the formation of glycogen in the liver and muscles, which is crucial for energy storage. In the liver, glycogen synthesis can utilize several substrates. The primary one is blood glucose, which is readily available and can be directly incorporated into glycogen. But the liver also has the ability to use other hexoses like fructose and galactose. These sugars are converted into glucose derivatives before they are added to glycogen, expanding the liver's capacity to store energy from different dietary sources. Interestingly, the liver can also utilize non-carbohydrate sources through a process called gluconeogenesis. Glycerol, derived from fat breakdown, and lactate, produced during anaerobic glycolysis, can be converted first into glucose and then into glycogen. This shows how versatile the liver is in maintaining blood sugar levels and energy reserves. In contrast, when it comes to muscles, the main substrate for glycogen synthesis is blood glucose. The muscles don't typically rely on other sugars or non-carbohydrate sources for glycogen storage, focusing primarily on glucose that comes from the bloodstream. This makes sense because muscles need quick access to energy during activity, and storing glycogen primarily from blood glucose ensures a ready supply during physical exertion. Understanding these differences helps us see how the body directs energy storage depending on the tissue and available resources, which is fundamental to overall metabolic regulation. Now, let's explore glycogen metabolism, focusing specifically on the synthesis of glycogen, also known as glycogenesis. This process is essential for energy storage, especially in liver and muscle tissues. When there's an excess of glucose in the bloodstream, the body converts it into glycogen for future use. The process begins with glucose entering the cell, where it's phosphorylated to form glucose 6-phosphate. This molecule is then transformed into glucose 1-phosphate, which is a key precursor for glycogen synthesis. The next step involves the enzyme UDP-glucose pyrophosphorylase, which converts glucose 1-phosphate into UDP glucose. This activated form of glucose is crucial because it provides the energy needed for the next step. Glycogen synthase, the main enzyme in glycogenesis, then catalyzes the transfer of glucose from UDP glucose to the growing glycogen chain, creating alpha-1, 4-glycosidic bonds. It's important to note that the formation of branches in glycogen, through the action of branching enzymes, increases solubility and accessibility for rapid energy release when needed. This entire process is tightly regulated by hormonal signals, primarily insulin, which promotes glycogen synthesis in response to high blood glucose levels. Understanding glycogen synthesis is vital because it highlights how our bodies efficiently manage energy storage, ensuring a quick supply of glucose during periods of fasting or increased activity. This process is a perfect example of the body's ability to balance energy intake and expenditure seamlessly. Now let's delve into how glycogen is synthesized, a process known as glycogenesis. First, glucose molecules are activated by converting them into a compound called uridine diphosphate glucose, or UDPG for short. This activation is essential because it makes the glucose more reactive and ready for incorporation into glycogen. Once we have UDPG, the next step is to add these molecules to a starting point, or primer, to build the glycogen structure. This primer can be either a few glucose molecules linked together by alpha-14 bonds or a special protein called glycogenin. Glycogenin acts as a scaffold, and UDPG reacts with a tyrosine residue on glycogenin to kick off glycogen synthesis. The enzyme glycogen synthase plays a central role here. It facilitates the addition of UDPG to the primer, extending the glycogen chain by forming alpha-14 linkages. Typically, this process elongates the chain by about 12 to 14 glucose units. But glycogen isn't just a straight chain, it has branches. That's where the branching enzyme comes in. It transfers segments of the elongated chain, usually 5 to 8 glucose residues, to a different part of the molecule, creating a new alpha-16 linkage. This branching not only makes the glycogen molecule more compact, but also increases its solubility and makes it easier to rapidly add or remove glucose units when needed. And this entire process is repeated, with glycogen synthase adding more glucose units and the branching enzyme creating new branches, resulting in a highly branched, efficient storage form of glucose that our bodies can quickly mobilize when energy is needed. Now let's talk about glycogen metabolism, specifically the breakdown process called glycogenolysis. This is essentially how our body mobilizes stored energy from glycogen. In the liver, this process releases glucose into the bloodstream, while in muscles, it produces lactic acid for energy use. The key enzyme here is phosphorylase. It acts on the alpha-1,4 bonds within glycogen, breaking them down through a process called phosphorylasis. That means it uses inorganic phosphate, or PI, to cleave the bonds, resulting in glucose 1-phosphate molecules. These are important because they can be readily converted to glucose 6-phosphate, which then enters various metabolic pathways. Phosphorylase works on the branches of glycogen that contain more than 4 glucosyl units. When it encounters a branch with exactly 4 glucose units, it can't cleave any further directly. Instead, the transferase enzyme shifts 3 of those glucose units to another branch, effectively extending that chain and leaving a single glucose unit attached by an alpha-1,6 bond. This is where the debranching enzyme comes in. It hydrolyzes that alpha-1,6 bond, breaking it down with water, and frees the remaining glucose unit. This process ensures that glycogen breakdown is efficient and continuous. Finally, the glucose 1-phosphate molecules produced are converted into glucose 6-phosphate by the enzyme mutase. This step is crucial because glucose 6-phosphate can then be used in glycolysis or other pathways, depending on the cell's needs. So, in summary, glycogenolysis involves a coordinated effort of enzymes to break down glycogen into usable glucose units, supporting energy supply during fasting or muscular activity. Now, let's take a closer look at what happens during glycogen breakdown, also known as glycogenolysis, and what occurs with glucose 6-phosphate afterward. In the liver, glucose 6-phosphate is converted into free glucose through an enzyme called glucose 6-phosphatase. This process is crucial because it allows the liver to release glucose into the bloodstream, helping to maintain blood sugar levels during fasting or between meals. In contrast, muscle tissue doesn't have the enzyme glucose 6-phosphatase. So, instead of producing free glucose, the glucose 6-phosphate in muscles enters glycolysis. This pathway leads to the formation of lactate, which can be used later for energy production or transported to the liver for gluconeogenesis. These differences highlight how the body efficiently manages glucose depending on the tissue and its specific energy needs. Now, let's take a closer look at glycogen metabolism and how it is regulated. It's important to understand that glycogenesis and glycogenolysis are tightly coordinated processes. When one is stimulated, the other is simultaneously inhibited. This regulation ensures that the body maintains proper blood glucose levels, depending on the metabolic state. During fasting, for example, glycogenolysis is stimulated to break down stored glycogen and release glucose into the blood, providing energy and maintaining blood sugar levels. At the same time, glycogenesis or glycogen synthesis is inhibited to prevent unnecessary storage. Conversely, after a meal, the situation reverses. Part of the absorbed glucose, about 40%, enters the circulation for immediate use by tissues. The remaining 60%, however, is converted into glycogen in the liver. So, after eating, glycogenesis is stimulated to store this excess glucose, and glycogenolysis is inhibited. The key enzymes controlling these processes are glycogen synthase, which promotes glycogen formation, and phosphorylase, which breaks down glycogen. Now, focusing on fasting, when energy is needed, hormones like glucagon and epinephrine come into play. They bind to specific receptors on cell membranes, signaling the need to mobilize glycogen stores. This binding activates a cascade involving CAMP, which then activates protein kinase. This enzyme is made up of 2 regulatory and 2 catalytic subunits. When CAMP binds to the regulatory subunits, it releases the catalytic subunits, activating them. This activation ultimately leads to the breakdown of glycogen, raising blood glucose levels or providing energy for muscles during exercise. So, in summary, this regulation ensures our body responds appropriately to different metabolic needs, maintaining energy balance across fasting and post-meal states. Now, let's look at how glycogen metabolism is regulated, starting with the activation of phosphorylase kinase. This enzyme has 2 forms, an inactive B form and an active A form. The transition from B to A is driven by CAMP-dependent protein kinase, which phosphorylates the inactive form, thereby activating it. This step is crucial because active phosphorylase kinase then plays its role in activating glycogen phosphorylase. Speaking of glycogen phosphorylase, it also exists in 2 forms, the inactive dephosphorylated B form and the active A form. When phosphorylase kinase is active, it adds a phosphate group to glycogen phosphorylase B. Converting it into the active phosphorylase of this activated form is what actually catalyzes the breakdown of glycogen into glucose units. Finally, it's important to note that the active phosphorylase can be turned back into its inactive form, phosphorylase B, by the action of protein phosphatase 1, which hydrolyzes the phosphate group. This regulation allows the cell to finely tune glycogen breakdown depending on the energy needs at any given moment. Overall, these phosphorylation and dephosphorylation steps are key control points that ensure glycogen breakdown is tightly regulated, linking signals like hormones to metabolic responses. Glycogen metabolism, specifically its regulation, is a complex but fascinating process. Let's focus on the inactivation of glycogen synthase, which is the key enzyme responsible for glycogen synthesis. Glycogen synthase exists in two forms, the active A form and the inactive B form. The critical difference between them lies in phosphorylation. The active form is dephosphorylated, while the inactive form is phosphorylated. When glycogen synthase is phosphorylated at multiple sites, it becomes inactivated, and the extent of inactivation depends on how many sites are phosphorylated. This phosphorylation process is carried out by several different protein kinases, which are themselves regulated by signaling molecules such as CAMP. Now, let's connect this to hormones like glucagon and epinephrine. When these hormones bind to their receptors on liver or muscle cells, they initiate a cascade that activates adenylyl cyclase through a G protein. This enzyme then produces CAMP, which activates protein kinase A once active. Protein kinase phosphorylates glycogen synthase, converting it into its inactive B form. Fortunately, this process is reversible. The inactive glycogen synthase B can be transformed back into the active A form by protein phosphatase 1, which hydrolyzes the phosphate groups. This regulation ensures that glycogen synthesis is finely tuned according to the body's needs, especially during fasting or stress responses. Understanding this regulation helps us see how hormonal signals precisely control energy storage, fitting into the broader picture of metabolic regulation we've been discussing. Uerove yelen yeri gilal ukawaze yeroveke erove ilbe yeruche, and opelove yerushe ayete esya. Ete aveste emu eru yeteve yerude yezelya en iya izen esu izene. Kawate a yerakratkoy. Iga era yete kewel uche yevegu u eral yeri ve yegizuel yetse. Iabe yelenage keoge yelena. Ienesu yena keteve yeruyete, efya yelente, fosfo de este yerase. Kalatel yerakratkoy per yeverash jayetitse animafam. Iatave janal. Kalatel muchatel amphes veluache de yeri. Espel asabes tevuyete, erazeru she yeleni uge keoge yelena. Kalogede atse, gumef umya en yeri shayete esya. Elef efya kate yege alena gilali keoge en o elize o esalebe yevayete. Iperotse ye ss eraze erushe ye en iya ge keoge ye en ezea emya de elya yete esya. En alete oyeshe ye en yeve esya. Ienesueliente akaje yakateri video yete efya emya ente efosfate vazo. Kalatel yerakratkoy udeo elya yete. Efosfate, en yeru yege eru papiyeru estevu kakil uche yeve yeruke efya emyelente ove. Gel keoge ye en esiente. Is a yeru keoge ye en afosf, o ariel is a yeru. Ete oache ye en yeri veje en yerakratkoy, imolim yelente. Pedatela muchate odea el ye en, ye osfate, en oakratkoy geru papiyeru akate videru yetege, keoge ye en esiente ezeu. Espel esabeestevu ye esiente, yezeuge keoge yelene. Yade en ove ye yelen en audien gedi bayer yetege keoge yezen fosf o ariel ezeu. Yelen yeri sha ya yegva kativigano este yeri. Kalake yezeu yeri taete. Yelen yeri sha yetege keoge ye en o elisa. Viditoge ye. Pedal ye de yeru. Bella gidodeo erio etelium yekalo zelama Paira yes kawadita sn ija yen. Iya era sjaya ya pedalya and ya galali kawagayana ya katetive eru yeta esya ya gawasian te yesa. Etela sn ove en o kratkoi, espera esabe. Kawatela yeru en ashaya or gera eniza, ya geu eli eru yeteze, apela es yeru en ya gedi vividi de ya ge, eli kawagayana. Pela de ya je ivea ya bea lmns, ye je debu ya ga, okelero in ya en em obe el izaetse, yelakratqui veze ave es honeste, idote pedote ya be en honeste yelakratqui. Glycogen metabolism and glycogen storage diseases are important topics because they help us understand how our body stores and uses energy. These diseases are a group of inherited disorders, meaning they are passed down through families. They are characterized by the abnormal storage of glycogen in tissues, either in terms of type or amount. The main cause of these disorders is a deficiency in one of the enzymes involved in glycogen metabolism. When these enzymes are missing or not working properly, glycogen can accumulate abnormally in tissues like the liver or muscles, leading to various health issues. There are 8 different types of glycogen storage diseases, each with its own specific enzyme deficiency. Today, I want to focus on the most common one, type I, also known as von Gierk's disease. This condition results from a deficiency of the enzyme glucose 6-phosphatase. In von Gierk's disease, because glucose 6-phosphatase is lacking, glycogen accumulates excessively in the liver. This leads to hepatomegaly, or an enlarged liver, and disrupts normal liver functions. Patients often experience fasting hypoglycemia, which means their blood sugar drops dangerously low during fasting periods. Additionally, these patients may develop ketosis and hyperlipidemia, reflecting how their bodies respond to low glucose levels by breaking down fats. Another key point is hyperuricemia, which can lead to gout. The deficiency causes an increase in glucose 6-phosphate, which stimulates the pentose phosphate pathway, resulting in increased ribose production. This, in turn, raises uric acid levels in the blood, leading to gout. Understanding this pathway helps us see how a defect in a single enzyme can have widespread effects, impacting energy metabolism and leading to these characteristic symptoms. This highlights the importance of proper enzyme function in maintaining normal metabolic processes. Now, let's take a closer look at some glycogen storage diseases, starting with type 2, also known as Pompe's disease. This is caused by a deficiency in the enzyme acid alpha-glucosidase, which is responsible for breaking down glycogen in the lysosomes. As a result, glycogen builds up inside cells, particularly affecting the muscles and heart, leading to muscle weakness and cardiomyopathy. Moving on to type 3, or Cory's disease, which is also called limit dextrosidosis. This condition arises due to a deficiency of the debranching enzyme, which normally helps in the complete breakdown of glycogen. When this enzyme is deficient, glycogen molecules have many short branches because they can't be fully debranched. This primarily affects the liver, muscles, and heart, leading to hypoglycemia and muscle weakness. Both of these diseases highlight how crucial proper enzyme function is for glycogen metabolism. If the enzymes involved are defective or deficient, glycogen accumulates abnormally, causing significant metabolic and structural issues in various tissues. This underscores the importance of the regulation of glycogen storage and breakdown in overall metabolic health. Today, I'll be talking about gluconeogenesis, which is essentially the process of forming glucose from non-carbohydrate sources. These sources include things like lactate, pyruvate, glycerol, some amino acids known as glucogenic amino acids, and in ruminants, propionate. Now, why is this process important? Well, gluconeogenesis plays a vital role in supplying the body with glucose, especially in situations where carbohydrate intake is low or glycogen stores are depleted. For example, glucose is the only energy source for nervous tissues, red blood cells, and skeletal muscles during exercise, so maintaining glucose levels is crucial. It also serves as the precursor for milk sugar, or lactose, in the mammary glands, which is essential for milk production. Additionally, during low-carbohydrate diets or after about 12 to 18 hours when liver glycogen runs out, gluconeogenesis kicks in to keep blood glucose levels stable. Another key point is that this process helps clear the blood of waste products from other tissues, like lactate, which is produced by muscles and red blood cells during activity. In terms of where this happens, gluconeogenesis occurs inside cells, specifically in the cytosol and mitochondria. Most of it takes place in the liver, about 90%, with the remaining 10% happening in the kidneys. So, overall, gluconeogenesis is a crucial metabolic pathway that ensures a continuous supply of glucose, supporting energy needs and metabolic balance, especially during fasting or intense activity. In this slide, we're looking at the major pathways and how they're regulated in the liver for gluconeogenesis and glycolysis. You can see the entry points for glucogenic amino acids after transamination, which are indicated by these arrows extending from the circles. These amino acids can serve as substrates for glucose production when needed. The key enzymes involved in gluconeogenesis are highlighted in these double-bordered boxes, emphasizing their importance in the pathway. It's also crucial to understand that the ATP required for gluconeogenesis mainly comes from the oxidation of fatty acids, which provides the energy necessary for this process. Now, an interesting point is that propionate, while important in ruminants, doesn't play a significant role in other species, so its contribution is more specialized. Moving on to regulation, you'll notice the arrows with wavy shafts represent allosteric effects, meaning certain molecules can regulate enzyme activity directly through binding. The dash-shafted arrows show covalent modifications like reversible phosphorylation, which modify enzyme activity more permanently. A key regulatory signal here is high concentrations of alanine. When alanine levels rise, they act as a gluconeogenic signal by inhibiting glycolysis at the pyruvate kinase step. This essentially shifts the liver's focus from breaking down glucose to producing it, especially during fasting or prolonged exercise. Overall, this slide highlights how tightly controlled and interconnected gluconeogenesis and glycolysis are, ensuring the liver responds appropriately to the body's energy needs. Let's take a closer look at the steps involved in gluconeogenesis. Essentially, most of these steps are just the reverse of glycolysis, which makes sense because we're synthesizing glucose from smaller molecules. However, there are 3 key steps that are irreversible in glycolysis, and these need special enzymes to be bypassed during gluconeogenesis. Focusing on the conversion between pyruvate and phosphoenolpyruvate, this process involves 2 particularly energy-consuming reactions. First, mitochondrial pyruvate carboxylase catalyzes the conversion of pyruvate into oxaloacetate. This reaction requires ATP and involves biotin as a coenzyme. Biotin's role is fascinating because it binds CoA2 from bicarbonate forming carboxybiotin, which then transfers the CoA2 to pyruvate, creating oxaloacetate. Now, oxaloacetate can't directly cross the mitochondrial membrane, so it gets reduced to malate, which is exported into the cytosol. Once in the cytosol, malate is oxidized back to oxaloacetate. The next step involves phosphoenolpyruvate carboxygenase, which decarboxylates and phosphorylates oxaloacetate to produce phosphoenolpyruvate, or PEP, using GTP as the phosphate donor. An interesting point here is that in the liver and kidneys, the enzyme succinate thiokinase produces GTP during the citric acid cycle, rather than ATP as in other tissues. This GTP is then used by phosphoenolpyruvate carboxygenase, creating a vital link between the citric acid cycle and gluconeogenesis. This connection helps prevent excessive depletion of oxaloacetate, ensuring that the citric acid cycle continues efficiently while glucose is being synthesized. So, in summary, these steps highlight how gluconeogenesis cleverly bypasses the irreversible reactions of glycolysis, and the interplay with the citric acid cycle ensures everything functions smoothly without depleting key intermediates. Let's look at some key steps in gluconeogenesis. First, consider the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. This step essentially reverses a part of glycolysis and is catalyzed by an enzyme called fructose 1,6-bisphosphatase. This enzyme is crucial because its presence in a tissue determines whether that tissue can make glucose not only from pyruvate, but also from triose phosphates. So, it highlights the tissue-specific capacity for glucose synthesis. Moving on, the next important step is converting glucose 6-phosphate into glucose. This reaction is catalyzed by glucose 6-phosphatase. What's interesting is that this enzyme is found in the liver and kidney, which are key organs in maintaining blood glucose levels. However, it's absent in muscle tissue, which means muscles can't export glucose into the bloodstream. Instead, they use it locally for energy. These steps are vital because they show how the body controls glucose production and release, ensuring a steady supply of this essential fuel, especially during fasting or low carbohydrate intake. Connecting this to the overall theme, understanding these pathways helps us appreciate how gluconeogenesis supports blood sugar regulation and energy homeostasis. Blood glucose levels are influenced by several key processes, including diet, gluconeogenesis, and glycogenolysis. When we consume digestible carbohydrates, they are broken down into glucose, galactose, and fructose, which are transported to the liver via the hepatic portal vein. In the liver, galactose and fructose are quickly converted into glucose, helping maintain blood sugar levels. Now, glucose can also be formed through gluconeogenesis, which involves two main groups of compounds. The first group includes those that can be directly converted into glucose, such as most amino acids and propionate. The second group consists of products from tissue metabolism, like lactate, which is produced during glycolysis in skeletal muscle and red blood cells. This lactate then travels to the liver and kidneys, where it is converted back into glucose. This cycle, known as the Cori cycle or lactic acid cycle, is essential for recycling lactate and maintaining blood glucose, especially during activity. During fasting, muscle tissue releases a significant amount of alanine, more than what is stored in muscle proteins. This alanine is produced by transamination of pyruvate, which itself comes from glycolysis of muscle glycogen. The alanine then travels to the liver, where it's transaminated back into pyruvate and used to produce glucose through gluconeogenesis. This process, called the glucose-alanine cycle, provides an indirect way for muscle glycogen to support blood glucose levels during fasting. The energy needed for this hepatic glucose synthesis comes from the oxidation of fatty acids, which supplies the ATP required for gluconeogenesis. Finally, don't forget that the liver can also break down stored glycogen directly through glycogenolysis to produce glucose when needed. So overall, our body has multiple interconnected pathways to ensure a steady supply of glucose, crucial for energy, especially during fasting or increased activity. Blood glucose, which is our main source of energy, actually comes from a few different sources. The primary one is the food we eat, especially carbohydrates, which are broken down into glucose. But our body also has mechanisms to produce glucose internally when needed. One of these is gluconeogenesis, which is the process of creating glucose from non-carbohydrate sources like amino acids and glycerol. And then there's glycogenolysis, which is the breakdown of stored glycogen in the liver into glucose. All of these processes work together to keep our blood glucose levels stable, ensuring our cells have the energy they need throughout the day. This balance is crucial for maintaining overall metabolic health and supporting our body's functions. Let's delve into the hormonal regulation of gluconeogenesis. First, we have glucocorticoids, such as cortisol. These hormones play a significant role by stimulating gluconeogenesis through a couple of key mechanisms. They promote the synthesis of enzymes necessary for this process, effectively turning up the production line. Additionally, glucocorticoids enhance protein breakdown in tissues, which increases the availability of glucogenic amino acids that can be used to generate glucose. Next, we consider glucagon. This hormone encourages gluconeogenesis primarily by lowering levels of fructose 2,6-bisphosphate. Since fructose 2,6-bisphosphate is an important regulator, its reduction shifts the balance toward glucose production. On the other hand, insulin acts as a counterbalance. It inhibits gluconeogenesis by preventing the synthesis of the enzymes involved, effectively turning down this pathway when glucose levels are sufficient. Moving on to allosteric effectors like acetyl-CoA and ATP, these molecules also regulate gluconeogenesis. They stimulate the process by two main actions. First, they inhibit glycolysis by inhibiting phosphofructokinase 1, which reduces glucose breakdown in the cell. Second, they promote gluconeogenesis by activating fructose 1,6-bisphosphatase. Specifically, acetyl-CoA has an additional role. It stimulates pyruvate carboxylase, an enzyme critical for gluconeogenesis, while simultaneously inhibiting pyruvate dehydrogenase, which shifts metabolism away from glucose oxidation. In summary, these hormonal signals and allosteric effectors work together to finely tune gluconeogenesis, ensuring glucose production matches the body's energy needs while maintaining metabolic balance. Gluconeogenesis, which is the process of creating glucose from non-carbohydrate sources, plays a vital role in maintaining our blood sugar levels, especially during fasting or intense exercise. When our body needs energy but dietary glucose isn't available, this pathway kicks in to ensure our cells, particularly in the brain, continue to receive the glucose they need to function properly. Now, an important aspect to understand is that gluconeogenesis is energy dependent. It consumes energy in the form of ATP and GTP. This means it requires a significant investment of energy to produce glucose, which makes sense because our body prioritizes efficient use of resources. Connecting this to the overall theme, gluconeogenesis highlights how our body cleverly manages energy and substrates to sustain vital functions, especially during times of scarcity. It emphasizes the balance our metabolic pathways maintain to keep us alive and active, even when external sources of energy are limited.